Hey everyone, welcome back for another deep dive. You know how this works. You give us the sources and we jump right in to extract the most interesting bits.
It's like panning for gold. We sift through the information and bring you the nuggets.
Exactly. And today we're diving into the microscopic world, specifically bacteria. You provided a really interesting flowchart about identifying gram-positive rods. It's a bit like a choose-your-own-adventure, but for bacteria.
Yeah, it reminds me a bit of a detective's guide. With each step, we narrow down the suspects until we have our culprit.
I love that analogy. So, ready to put on our detective hats?
Let's do it.
Okay, let's take a look at our first clue. At the very top of the flowchart, we see gram-positive rods.
Right. That's our starting point. These are bacteria that stain purple in a gram stain test and have a rod-like shape under a microscope.
So we're already narrowing down the field.
Mm.
Our source mentions Bergey's Manual of Systematic Bacteriology, which is like the ultimate encyclopedia for bacteria.
Yeah, it's the go-to resource for identifying and classifying bacteria. But this flowchart gives us a nice simplified roadmap within that massive world.
Okay, so we've got our gram-positive rods. What's the first question we need to ask?
The first question on the flowchart is spore forming.
Spores. Okay, remind me, what are spores exactly?
Well, picture this. The environment gets tough, resources are scarce, things are just not looking good for a bacterium. Some bacteria have this amazing ability to basically hit the pause button on life.
Wow, really?
Yep. They form these tough, dormant structures called spores that can survive all sorts of harsh conditions. Heat, dryness, even radiation. Then, when conditions improve, the spores germinate and the bacteria come back to life.
That's incredible. So it's like a bacterial survival strategy.
Exactly. It's an incredible adaptation that allows some bacteria to survive for incredibly long periods, even in the most extreme environments.
So, like back to the flowchart, which bacteria are we talking about here?
Yeah. Well, if we follow the plus path on the flowchart, meaning the bacteria do form spores, we're led to some familiar names, like Bacillus and Clostridium. You probably heard of them.
Yeah, Clostridium. That rings a bell. Something to do with food poisoning.
You got it. Clostridium botulinum, for example, produces a very potent toxin that causes botulism, and guess what? It forms spores.
Oh, so those spores are the reason why it's so hard to get rid of those dangerous bacteria in food.
Exactly. Spores can survive boiling temperatures, which makes them a real challenge in food safety.
Wow. Okay, so going back to the flowchart, what if the bacteria don't form spores?
Then we follow the 8 path, which leads us to the next question, acid-fast.
Acid-fast. I'm not familiar with that one.
It's a special staining technique. Basically, some bacteria have a unique cell wall structure that makes them really good at holding on to a certain dye, even when you try to wash it away with acid. Hence the name acid-fast.
So it's kind of like a stubborn stain that just won't come out?
Exactly, and one of the bacteria that's acid-fast is Mycobacterium smegmatis.
Mycobacterium. That sounds familiar too. Isn't that related to tuberculosis?
You're thinking of Mycobacterium tuberculosis, which causes TB. Mycobacterium smegmatis is a different species, often found in soil and water, and it's generally not harmful.
Got it. Okay, so we've got our spore-forming bacteria like Bacillus and Clostridium, and our acid-fast bacteria like Mycobacterium, and all this just from 2 questions on our flowchart.
Pretty neat, huh? It's amazing how much we can learn about a bacterium just by asking the right questions.
Absolutely. So what happens next on our bacterial adventure?
Well, let's say we're on the spore-forming branch of our flowchart. The next question asks strict anaerobes.
Meaning,
can they survive in the presence of oxygen? Some bacteria need oxygen to live, just like us, but others, particularly certain Clostridium species, can't tolerate oxygen at all. In fact, oxygen is toxic to them.
so they're like the opposite of us. We die without oxygen, but they die with it.
Exactly. They're called obligate anaerobes because they're obligated to live in oxygen-free environments.
Fascinating. Okay, so that's another fork in the road on our flowchart. What's the next test?
The next one is the catalase test.
Okay, what's that all about?
Catalase is an enzyme that breaks down hydrogen peroxide, you know, the stuff people use as a disinfectant.
Yeah, it fizzes when you put it on a cut.
Exactly. That fizzing is the catalase enzyme in your cells breaking down the hydrogen peroxide. And the same thing happens with bacteria. Some bacteria produce catalase and others don't.
So how does that help us identify them?
Well, if a bacterium is catalase positive, meaning it produces the enzyme, it'll bubble when you add hydrogen peroxide. If it's catalase negative, nothing happens.
So it's like a little chemical test to see if they have this special enzyme.
Precisely. And this helps us differentiate between certain types of bacteria. For example, Corynebacterium species are catalase positive, while Lactobacillus species are catalase negative.
Got it. So we're getting more and more specific with each step. What other tests are there on this flowchart?
Well, we have a test for starch hydrolysis.
Starch hydrolysis.
Basically, it's a test to see if a bacterium can break down starch. Starch is a complex sugar found in plants, and some bacteria have the enzymes needed to digest it.
So it's kind of like, can they eat starch?
Exactly. And this ability, or inability, to digest starch helps us tell apart closely related species. For instance, Corynebacterium kutcheri can hydrolyze starch, but Corynebacterium xerosus can't.
Wow, so even subtle differences in their diet can help us identify them. That's amazing. Are there any more tests on this flowchart?
We have a couple more. There's glucose fermentation activity.
That sounds a bit more complex.
It's not too bad. Basically, different bacteria ferment glucose, a simple sugar, in different ways. Some produce acid, others produce gas, and some produce both. By looking at the byproducts of fermentation, we can identify specific lactobacillus species.
Ah, so it's another way to distinguish between those closely related bacteria and the last test on the flowchart.
The last one is mannitol utilization. It's similar to the glucose fermentation test, but it uses a different sugar, mannitol. Again, this helps us differentiate between closely related lactobacillus species.
Okay, so we've gone through this whole flowchart using a series of tests to narrow down the possibilities and identify specific bacteria. It's like a detective story, but at the microscopic level.
Exactly, and it's not just about classification for the sake of it. Identifying these microbes is crucial for understanding their roles in the world around us, from the soil to our own bodies.
It's like once we know who they are, we can figure out what they do.
Right. For example, knowing which bacteria are causing an infection helps doctors prescribe the most effective antibiotics.
So bacterial identification has real-world applications in medicine, food safety, and all sorts of other areas.
Absolutely. It's a fundamental part of microbiology.
This whole deep dive has really opened my eyes to the fascinating world of bacteria and makes you wonder, what about all the bacteria that haven't been identified yet?
That's a great question. There are likely millions, maybe even billions, of bacterial species out there that we haven't even discovered yet. And scientists are constantly developing new techniques to identify and characterize them.
It's like a whole hidden universe just waiting to be explored. Who knew there was so much going on at the microscopic level?
That's the beauty of science, isn't it? There's always something new to discover, even in the tiniest corners of the world.
Absolutely. Well, thanks for joining me on this bacterial adventure. I feel like I've learned so much.
My pleasure. It's always fun to dive into the microscopic world.
And to all our listeners, keep those sources coming. We'll be back with another deep dive soon.